Dear friends, dear colleagues, dear ladies and gentlemen, my name is Miklós Kelemeyer. I am a professor at Semmelweis University and the dean of the medical school here. And it is my great pleasure and honor and privilege to host this session, which is a highlight of the past 2 days. And because this is an opening of a lecture series, and this event marks also the first-year anniversary of the start of a prestigious European University grant program, which is named EUniWell, a short word for European University for Wellbeing. As I mentioned, this is a highlight because together with us, we have Professor Botond Roska from Basel, Switzerland, my good friend, and I feel honored that he accepted our invitation to give us a talk and a lecture on his current research. Let me give you just a background information on this grant program. As the name implies, the topic of this and the target and the objective of this program is well-being, whatever that word means. I'm sure everyone has a connection to this word. And there could be nothing better topic for today to discuss what well-being is the loss of sight, loss of vision. And Professor Roska's research is aimed at restoring vision, as the title of his presentation implies. The past 2 days have been spent in a program called FestiWell, which is a festival-like commemoration of the first-year anniversary of the start of this grant program. And this program will be going on for 2 more years, and hopefully maybe for another few years as well. So this moment is the highlight of this FestiWell program. And with that, I'd like to introduce to you Professor Botond Roska. He's the director of the Institute of Molecular and Clinical Ophthalmology in Basel, Switzerland. And I must acknowledge the degrees that he has. After all, he's a graduate of Semmelweis University, so he's on one hand a physician, but at the same time, he became a mathematician, and he's also a neurobiologist, a geneticist, and believe it or not, he's also a cellist. So he has very high-ranking knowledge and skills. He has received a number of prizes. I just want to mention a few of the most important ones. A couple of years ago, he received the Order of Saint Stephen, which is the highest ranking honor in Hungary. He received the Louis Jeantet Prize in Medicine and the Kerber European Prize as well. He has been nominated a few times for the Nobel Prize, I should mention that. And by the way, he's also the recipient of our university's highest award, the Semmelweis Budapest Award. As I mentioned, the title of his presentation is Restoring Vision, which is based on his current results that I'm sure he will expand and explore. But in the recent years, he has made significant discoveries which carried him en route to the possibility that is in his hands, the possibility of restoring human vision, restoring the status of the loss of sight. He has described an atlas of cell types, neurological cell types in the retina. He has uncovered or discovered retinal circuits, and he discovered a developmental switch in the retina as well, which led him towards cell type targeted approaches, which allows him to make a significant step towards restoring vision. Having said all this, it is once again my utmost pleasure and privilege to have you with us, Botond. So please, the floor is yours.
Thank you very much, Miklós, for this very kind introduction. Let me share my screen with you. Do you see my screen?
Yes.
Very good. And I start my presentation. So thank you very much. And I like to continue this theme of well-being for humans. So vision, which I'm studying, starts with a video. and that video enters our eyes. It's captured by the retina. And then from here, it starts a journey towards the brain. One of the most important first station is the lateral geniculate nucleus in the thalamus. And then from here, information arrives to the primary visual cortex, and then it radiates through the human cortex. Indeed, around 50% of the human cortex, we can measure visual information. So humans are very highly visual. And this is reflected in a recent questionnaire in the US, when they asked people to name the worst conditions from a list of not very good conditions. And blindness was named #1, ahead of Alzheimer's disease and cancer and AIDS. And you can wonder, why is blindness so important for today? I mean, one reason I mentioned to you, which is the human brain is very highly vision dominated. But a second important notion here is that over the last 10 to 15 years, we became almost entirely dependent on vision, using our cell phones and small computers for not only work, but for social activities, especially during COVID time. You probably realize that all your social activities have been using digital devices and your eyes as a primary sense for gathering information. So the retina is a tiny part of the visual system. And its importance lies in the fact that almost all visual diseases which are causing blindness are localized in the eye, and especially due to problems in the retina. Now, the retina is a biological computer. It's doing image processing. There is one video that enters the retina via its photoreceptors, but then around 30 different movies that are created by the retina. And each of these movies is a feature that the retina extracts from the visual scene. And these 30 different features, such like lateral motion, approaching motion, different colors, are simultaneously broadcasted towards the brain. If we zoom into the retina, what we see, this is a drawing from Ramon y Cajal, so this is already solved by Ramon y Cajal, is a lot of different morphologies, a lot of different shaped cells. And Ramon y Cajal's notion, an important notion, was that there are approximately 100 different shapes in the retina, and each of these shapes are repeated in many times. This is an idea of a cell type. Indeed, later turned out that all our organs are made of many cell types. And this is the elementary unit of computation within the retina. So over the last 100 years, people studied how these cell types give rise to the functional architecture of the retina, how these movies are extracted by the dynamic interaction of these cell types. We studied disease mechanism based on cell types. And finally, recently, we and others started to develop therapy, cell type-based therapies. Now, the general architecture of the retina is conserved all the way across vertebrates, from fish to humans. However, our details are different. And these details are important enough for disease that in most animal models, the disease mechanism are different. So we cannot reproduce the human disease in animal species. Turns out the theory of development also is misleading when we do it in animal models. Therefore, it became extraordinarily important to develop therapy and understand disease in the context of the human retina. And so today, what I will talk about is therapy development. I like to show you a new model system, human model system, how we develop therapy. And then at the end, I will focus on one particular therapy that I've been developing since 20 years and show the results of the first patients that we treated. And all has to do with restoring vision in blind patients. So the disease I will be focusing today is retinitis pigmentosa. It's a disease that's causing blindness. It's a disease of the photoreceptors. And then usually patients start to lose their vision at the age of 30 to 40 and may become completely blind by the age of 50 to 60. The interesting thing about this disease is that although the photoreceptors lose their ability to detect light, the rest of the computer, the rest of this biological computer that's doing the processing remains intact. So the cells that compute information are there, but there is no visual information. And around 19 years ago, when I was a graduate student in Berkeley, I had a very simple idea. At that time, people started to publish sensors, light sensors from algae and bacteria. And the idea was to take a light sensor from algae, which are very simple, as opposed to human sensors, which require many components. These are simple sensors, and somehow build them into the retina, into the remaining computational element to resensitize the retina to light. And of course, the retina, as I told you, have around 100 different cell types, and we came up with key nodes, key cell types in which we can target these light sensors. For example, we can target them to remaining cones, which are light insensitive. We can target to so-called bipolar cells, particular type of amacrine cells, or the ganglion cells, which are the last processing elements of the retina. The ganglion cells from the optic nerve, they connect the retina to the brain. And over the years, we have worked on many of these different approaches. But what is common to all of them is the problem of how in a living human, we can target one specific type of cell and not the other 100. And so what we came up around 10 years ago is an idea to take gene therapy vectors. These are little balls that we can inject to the eye. These are protein balls, which has a DNA in it coding, for example, for the light sensors. And we came up with an idea of a so-called artificial promoters. This is small DNA segments that we try to find that instruct the gene therapy vector to be expressed only in a particular cell type. And I don't go into the design, how we make them on our computers, but just that how can we test them? So once you make a library, let's say 250 of these such gene therapy vectors, you have to test them in human retinas, because I told you that animal retinas, a number of gene therapy vectors and other therapeutic vectors behave differently. So we have really developed two different approaches. The first approach is to build human retinas in a dish. And this starts from skin biopsies or from a blood draw, where we reprogram either fibroblasts or lymphocytes into iPS cells. And from here, we started to use a procedure developed first in Japan by Yoshiki Sasai to start to grow optic cups from a cell, a mass in these so-called embryonic bodies. And from these cell mass, you can see this outgrowth of these optic cups. However, as we found, these optic cups are very primordial retinal-like structure. So over 10 years, we developed the procedure from this very primitive structure to make human retinas that has very similar structure. We call it organoids, retinal organoids, very similar architecture than that of the normal human retina. So on the left, you see a retinal organoid, on the right, a human retina, and you can see that both of them has 5 layers, and they have like the normal human retina, and they have the connectivity and a structure that you can see in the human retina. Now, these organoids actually also have many of the cell types that we can see in a normal human retina, but moreover, the cells, we saw connectivity between the different cells, suggesting that they might actually process information as a normal human retina. These are called synapses between neurons. That's what we can see here. So we wanted to test whether these retinas that we built in dish, are they responsive to light, and if they're responsive, can they talk to each other? So we can use, again, gene therapy vectors to bring in calcium sensors. So activity sensors, genetically encoded activity sensors in these organoids and can shine light to them and show that indeed you can see these response curves on the right. As we shine light, we can see this decrease of activity in the photoreceptors as in a normal human retina. And you can see that the amplitude is decreasing as it adapts to like a normal human retina. Moreover, we can go to second order and third order cells. So these cells that receive normal information, photoreceptors, and record from them and again see an activity signal, which we can completely block by blocking the communication between neurons, so-called synaptic blockage. Now, one particular problem in this is that I told you that we have 250 viral vectors, and by nowadays we have many thousands of these gene therapy vectors we want to test. So we needed to develop these in very large quantities. And Magdalena Renner, at that time in my lab, developed the procedure, a new procedure. How can we make essentially from one plate, a 6-well plate of cells, 22,000 retinal organoids? So we now have essentially tens of thousands of retinal organoids we can make, and we can screen now any number of vectors in them. And I just like to show you, so we can put them into 96 well plates. We are sort of building the robotics right now, so we don't have to do it ourselves. All the sorting and the growing is will be soon by robotics. And then we can screen vectors, and here is one hit, for example, where out of the 100 cell types, one of our vectors is only labeling human cones. Now, this method, we can screen a lot of retinas in a dish, but this is still not a retina of a 60-year-old patient. This is a retina we have built, so we have absolute certainty that these vectors will target the cell types in an adult human retina. And this is when we turn to another technology, namely to keep real human retinas alive after death. And this technology was developed mostly by my collaborator, Arnold Szabó in Budapest. So Serge Picot initially built a method to keep human retinas postmortem alive for 3 weeks. This is not yet enough to have expression from gene therapy vectors. And Arnold Szabó at Semmelweis University, he mastered this approach that he can keep alive human retinas for 14 weeks. So we started to collaborate with Arnold. We sent him hundreds of AVs via post from Switzerland to Budapest, where he applied these viruses, and we analyzed together. And what we see is with complete precision, some of our vectors can target, for example, ProA7 targets human cones with exclusive precision. We can see another vector targeting human rods, horizontal cells. I can name on. The more vectors we try, the more we can target, the more different cell types we can target. And we hope that in a few years, we will be able to target all human cell types just by this logic of artificial promoters. Now, I'd like today to focus on ganglion cells. We have 4 approaches, as I mentioned to you, to do a vision restoration using algal sensors. And in the first patients, we went for the simplest approach, the ganglion cells, in which the retina will completely lose computation because the ganglion cells are the last cells in the retina. The ganglion cells, we make them light sensitive, hoping that the patient are able to use some of these new vision that's coming from ganglion cells to conclude something about the visual scene. Now, this took a long time, around 15 years. It's a collaboration between José Sahel, Serge Picot, and Gensight. It's a small company. We started in Paris, and we went all the way from mouse studies to human ex vivo retina studies, to primate studies, and finally to clinical trials. This therapy we are using is a so-called 2-component therapy. One, we have a gene therapy vector that we inject to the eye. You need a single injection because these gene therapy vectors express their cargo for a very long time, likely until the rest of the life of the patient. But we also need a goggle, and I will explain soon why we need a goggle. But I just told you that ganglion cells are not the normal cells that sense light, and a lot of computation is lost. So these goggles have a camera facing the world. It has a computer interface, and then a projector that projects something to the eye. So here you can see the computer, which is in the pocket of the patient. Outside is a camera. Internally, here you can see the projector that projects to the eye. So why do we need goggles? So the most important point is that the dynamic range of optogenetic sensors, these algae sensors called optogenetic sensors, are alone 2 logarithmic units. So for example, they can sense, let's say, 100 photons up to 10,000 photons, or 10,000 to 1,000,000 photons. But so they have a 2 log unit dynamic range, but the dynamic range in the visual world is around 10 log units. So 1 photon to many billions of photons we can see. So we need somehow a camera that can adapt to the right range, and then we can channel or tunnel these 2 log units to the sensitivity range of the sensor. The second one is that these algae sensors all have a particular wavelength sensitivity. So we want to have most of the power that we radiate in there at the relevant wavelength. And finally, I mentioned it earlier, we need computation. We need to mimic the computation the retina would perform, since some of the retinal computation is lost. So I like to tell you how a normal retinal ganglion cell will respond to light. These are recording from 5 different ganglion cells in the retina by stimulating them with a naturalistic stimulus. And what you see that the ganglion cells under natural vision, they produce these spike trains. So these activity bouts they have, and otherwise they are silent. So this is what we mimic with our computation. Our goggles produce around 60 millisecond local light pulse when the camera detects a change of intensities. So this is a very special camera that would not respond when there's no change of intensity, but as long as, for example, a bird would come by, those pixels, when there's a change of intensity, at that pixels, there would be a flash on the retina locally. Or, for example, you will see when I show you the patient, is that a patient will scan the scene and is doing so in order to evoke changes of contrast if the object is standing and not moving. Finally, we used a light sensor from algae called Crimson R, and there are thousands of algae sensors. So why do specifically we use these sensors? And one of the reasons, it's a particular sensor which is red-shifted. It's around 590 nanometer absorption maximum. And this is important because most of these optogenetic sensors are around 470, 500 nanometer, and there is a massive decrease of phototoxicity if we use 100 nanometer red-shifted light. So in this case, around 600 times less phototoxic if we use red light sensors. We also look at exactly how fast are these sensors. We use try to use fast sensors because, as I told you, that we have cameras that detecting change. If a car would come by, if you would use slow sensors, you would have a so-called motion after effect. Basically, you would confuse moving stimuli. But at the same time, we have to make sure that the sensor is not too light sensitive, because we didn't know at the beginning, for example, if we get any side effect in patients. You can imagine that maybe these patients get a headache if they get a new vision. So we wanted to make sure that we can turn off the stimulation. In this case, you can just simply take off your goggles, and in a room environment, you will not get stimulated. And finally, I just like to mention something that by today, there are likely better sensors that we are using here, but in a clinical trial, usually around 5 years before the trial, you have to fix your sensor and go through all the FDA and EMEA applications, safety applications, to be able to use for humans. So what I will show you now is our first patient, and we have a few more now in Paris and in the United States. So I will show you a video, and in this video, there will be two conditions. There will be a patient who will have with the goggle or no goggle. The injection is unilateral, so one eye is injected, the other eye is covered, or sometimes both eyes are serving as controls. And the first test, we ask patients to detect an object on a table. So the three tasks is perceive the object. So the person will say, well, I see something. And then to locate the objects, and then to touch the object. So the location is a verbal description that it's on the right, or something like that. And finally, the patient has to touch the object. And one important notion that these patients there are usually no patients who are completely blind. They have a so-called light sensitivity that they cannot tell any objects apart. You will see that. But like amoebas or very simple organism, they can say either light or no light. So that's the level of vision they are. And what we are aiming is to bring back object vision. So let me show you the first video. It's a French patient. You will see with English subtitle. Natural monocular means there is no goggle, one eye covered. So the injected eye is not covered. And the task is to find this object on the table.
Je perçois le bord de la table, je perçois. Au bout d'un certain temps, je ressens qu'il y a une différence d'intensité. Mais sur la table, je ne vois pas.
So he's unable to see anything. And now we put on the goggle. So pay attention on this camera at the bottom, which shows what actually the camera sees on the goggle. So not the camera, I'm sorry, the computer. So pay attention on the computer monitor, and you will see what the camera sees. So first of all, he's scanning. So see, his strategy is to scan. That is to detect changes in contrast.
Okay.
Now he perceived it. You can see it's on the computer.
Okay.
Now he located it. And he finds it. Okay, the next I show you a little bit more complex. We ask the patient to count objects, smaller objects on the table. So again, what you see now is natural binocular. That means no eye covered. This is to control that if he can see anything.
Qui a un truc devant moi. Mais c'est non, je ne peux pas.
Now we put on the goggle. See, now one of them appears. Watch how he's scanning. It's important, he's also holding to the table. Thereby he knows what is the area he's searching. Is one. And another one.
Là, j'ai une deuxième qui est pas le.
So he realizes there are two.
J'en ai un deuxième. D'accord. Qui est sur la première ligne, qui est sur la droite.
Then he describes the relative location of the two objects. So he says on the same line there are two. And finally, I like to finish with the final test where we wanted to test the limit of this patient. So we decided that we show a hydroalcoholic gel, which is almost transparent. Again, this is a control condition, no goggle.
Non, je ne vois pas.
Now we put on the goggle.
C'est dingue. Je me mets à rechercher une vibration.
Very interesting that he sees vibration and there's an object. We don't fully yet know what is this. We are testing. So what's important here is that he really takes time to search for it. There's no goggle, and he sort of systematically goes through and concludes there is nothing. And then now with the goggle, he sees almost immediately. He finds something. He describes the location.
Et ben, c'est exactement ça.
Voilà, c'était là. Alors, j'ai essayé de le pointer. Je suis passé sur la distance. Là.
Okay, so clearly the depth location was not correct, but he sees it, so he keeps searching for it. So these were 3 behavioral tests, but we also wanted to have an objective evidence that the brain signals are representing the objects. So what we have done is a test in which there was an object or no object, a recording with EEG from many different locations in the brain. And what we could do is first see that in the occipital electrodes, we can find a signal representing the object. And then purely using this signal, we could decode the presence or the absence of the object with 80% precision. Now, the last video I'd like to show you is about well-being. We heard from Miklos is that this conference about well-being. And in medicine, when we design a new therapy, the question is that, okay, this person is able to do some detection. Is it good for the person? Is it helping the daily life of that person? And regulators, when we go for asking this treatment to be a medicine that can be given to in Europe or the US, the primary question, does it increase the quality life of the patient? And this is what I'd like to finish, is that you will see a very surprising, for us, very surprising quality increase when the patient says he loves to cook. Well, he's French. And now he can start to cook again. And this is an interview with the patient.
Avec vous la façon dont vous avez appris votre diagnostic.
Alors, j'ai appris ça, j'avais 26 ans. Et mes parents m'ont envoyé voir un ophtalmo, naturellement. On a corrigé une petite myopie, si j'ose dire. Et quelques années plus tard, je suis retourné à Paris voir un ophtalmo, et c'est à ce moment-là qu'il m'a appris que je n'étais pas que myope. J'avais sans doute une rétinopathie ou une rétinite pigmentaire. Mais à un moment, je me suis quand même retrouvé sur les rails du métro. Et là, j'ai un médecin qui m'a dit, bah, écoute, tu vas t'arrêter, je vais t'arrêter de bosser parce que tu vas te tuer. Ah. Et voilà, et de ce jour-là, j'étais en vacances.
Qu'est-ce que vous apporte cet essai clinique?
Le premier sentiment, quand j'ai effectivement eu le système sur les yeux et la première fois que ça a fonctionné, et quand je dis la première fois, ça a fonctionné dès la première fois. C'est assez impressionnant. À un moment, j'ai souvenir, dans une des expériences qu'on a pu faire, où je me suis retrouvé dans la rue à chercher les passages protégés. Et il s'avère quand même que j'ai trouvé les passages protégés alors que j'avais le device sur les yeux, et que je ne l'ai pas retrouvé quand on a refait l'essai juste après sans les lunettes. C'est assez impressionnant parce que ça a excité quelque chose dans l'œil. Je me suis dit non, je ne vois pas, c'est mon cerveau qui voit. Ça donne des images qui sont très très particulières. Quand je dis que je vois les choses scintiller, que la perception, c'est de la lumière qui vibre.
Mais justement, qu'est-ce que vous envisagez quand vous allez avoir la possibilité d'utiliser les lunettes chez vous? Est-ce qu'il y a des moments, des circonstances où vous allez peut-être choisir plus de les utiliser? Oui,
alors j'ai une idée, c'est effectivement quand je fais la cuisine. À un moment, je réussis à trouver vous savez, la table à induction. Pourquoi? Parce qu'elle est noire sur un fond blanc. de continuer à m'exercer, de voir si ça progresse, de voir si, entre guillemets, ça s'améliore, et ensuite de s'exercer pour s'approprier le système. En fait. Quand je mesure le résultat pour moi, et pour quelqu'un qui, comme moi, avait fait le deuil de sa vue, si j'ose dire, c'est quand même un truc assez extraordinaire, un moment de se dire, ben voilà, je revois. Voilà, c'est plein d'espoir.
So, question is, what did we learn? So this was the first patient. Now we have a few patients with similar level. And I would say the first thing that we learned is that we gave back a sensation, but it's a new sensation to the patient. It's not exactly the same vision as before. As I said, there's no retinal computation. The ganglion has passed a different kind of message than normal. And what was really interesting to observe in this patient, that what you're seeing is a learning curve. So the patient gets this new kind of vision, and over months, he's learning more and more about the visual scene. It's like a child learning a new language. There's a new information coming from the world, and he's learning to interpret it. So it's a learning exercise and an interplay of memory and learning these new ideas, putting them with previous ideas he had about object, and forming a new coherent picture about the visual world. So that's number one, very important, because the most important part of the therapy is not the injection, because that's a surgical injection that takes an hour or less, but the most important is a rehabilitation program. We don't know how to rehabilitate this patient. We are learning to rehabilitate. So what we can improve the resolution, for example. We can improve the computation. We can change the computation in the device very easily. And we constantly have to ask the patient what is bringing better and better vision. And we can improve the learning exercise that we are providing to the patient. Moreover, in the future, ganglion-based approach is one approach. And now in Basel, we are bringing a different approach, cone-based optogenetics, to the clinic, and hope in 5 to 10 years, we will have 4 different approaches, or 3 to 4 different approaches in clinical trials. And we can particularly say for a given type of patient, which cell types we would like to target, which one possibly bring back the largest amount of visual capability. We will never be able to restore vision that the person will be able to read the New York Times in its original way. But I think that we can be able to bring back vision that is increasing the well-being, the quality of life of these patients. And we hope to do so that more and more vision can be restored as we refine our technology. So I'd like to say thanks to a lot of people who worked on it. Some people from my lab, Josephine Jüttner, Cameron Cohen, Magla Renner, and Stefan Spirig. My co-director here in IOB, Hendrik Scholl, and José Sahel and his team in Paris, with whom the ganglion cell-based visual restoration we did together in the last years. So I'd like to thank you very much for your attention. And again, thank you very much, Miklos, for the kind invitation.
Thank you so very much, Professor Roska. Please accept our heartfelt acknowledgement and thankfulness towards this fantastic, fascinating lecture. It is truly was a highlight, embedded very much into the concept of well-being. As you well pointed out, that loss of vision is the one that's associated with the major loss of well-being status. If we may, there were a few questions that arrived through the chat, and you can see them, by the way, as I think, but there were a number of questions by Lars Brandén, and the first one by him was, does the fact that the eye is an immune privileged organ matter in your work, and how you focus on finding a cure for any specific eye condition? I think you pretty much answered it in your talk, but if you want to reflect on that.
Yes, so it does matter, of course. The brain and the eye are immune privileged. In the eye, you can do so-called subretinal or intravitreal injection. This was an intravitreal injection. The subretinal space is even more immune privileged, but we didn't see any major immune problem here, although we used a sensor from algae. And the question for, can we find a cure for any specific eye condition? I think that so far we worked on complete blindness. And I would say in complete blindness, because we have light sensor, as long as there's an optic nerve, we can definitely find a way of restoring some amount of vision independent of what was the cause. Now you go to diseases which are not complete blindness. We cannot use the same approaches as I've shown here, because we actually project quite a large amount of light, and if you have remaining vision, that would not work. And here we are working on a completely different approach using near-infrared light that is not visible to humans to project. So that's a next step. But there are a lot of other conditions, eye conditions, for example, for glaucoma and diabetic retinopathy. There's absolutely no cure today, and we don't even understand the disease. One of the problems of many of the eye diseases, for what we call the large eye diseases like macular degeneration, glaucoma, diabetic retinopathy, that we have absolutely no clue about the cause of the disease. And this is very surprising, given that there are hundreds of million patients around the world. The problem is that, as I mentioned to you, that the primate eye in evolution is become very different from the other mammalian species, because we have a fovea that is at a high resolution area of the retina, which is entirely missing in other mammals. So we have to really study the human retina. And this is something that we started to do in collaboration with Semmelweis University in my institute, but all diseases is really, especially when we have remaining vision, is a big task.
Well, this is truly fascinating. There are a couple more questions from Lars Brandane. Let me read the second one. Do you have an issue with the purification of infectious, that is, filled AAV particles from the empty AAV particles, and what is your approach to maximize the amount of functional versus non-functional AAV particles?
Yeah, so that's really, it's a very excellent question. It's a technical question that the methods we used here, there is around 90% field particles, but there are better methods to purify them, which you get more pure AAVs. Then there's a problem, you get less field particles. There are a number of approaches now used by industry, for example, antibodies that you can pull down, for example, field particles versus empty particles. I don't know actually why they pull down, no one understands it, but you can separate them. And there are several other ways of separating them. It's all very costly. It is quite costly to, unfortunately, purify gene therapy vectors.
Thank you very much. Yet another question in this line. Are you also exploring the various serotypes of AAV for specific cell targeting, or relying on synthetic and specific promoters exclusively?
So serotypes never give complete specificity, at least not the one that we have now. So we do explore always. So we have all the serotypes available today for the community, and we test all of them. But this gives you a broad, it's an availability. It never gives you specificity. It's not impossible to make specificity. For example, the very recent method to put nanobodies, which is small binders, on the surface of AAVs to have some specificity of entry. And we are exploring that, but this is not yet in clinical practice.
Well, thank you very much. And the final question from Lars. Are you working on different version of the light receptors for different wavelengths?
Yeah, very good question. In principle, we can bring back color vision using the approach, and the answer is yes. We are working on it, but this is still basic research and not yet therapy development.
Thank you very much, Botond. May I ask a couple questions myself, simply because I was so inspired by talk. The first one is you showed these beautiful organoids, which had this layered structure. I was just wondering whether these layers occur or develop spontaneously, or do you need to, do you have some trick to do that, to achieve that?
Completely spontaneously. So our trick, so it was actually very interesting to discuss, is that what did we do differently than others, that our became layered and the others didn't produce. And what we found is that what matters really the initial condition. In other words, that when we call them iPSC cells, each of them is somewhat different. These are not pluripotent stem cells. They are different states. And indeed, we worked around 5 years to try to use different cocktails to make layered retinas. We never succeeded, no one else. And the idea how we built actually the layer structure was coming from mathematics, when one day I realized that the problem is very similar to nonlinear dynamics, when we know that what qualitative solution you get at the end really depends on the initial condition. So from there on, we thought that we vary the initial conditions, namely the iPSC lines, or we reprogram several times. And then we found some initial conditions that make very good retinas. Now, what is exactly this initial condition? We don't know. But for example, when you reprogram, you use a Yamanaka factor in unknown concentration. So there's a lot of different, you are in a 4-dimensional space, you search around, and we just find some very good initial conditions that gives us. So the strategy we use now is we have few lines, cell lines, that creates these multi-layered retinas, and we edit in mutations. For example, if you want to study disease.
Right, and so is the synaptic connectivity also will be mimicking the retinal one?
That's correct. The synaptic connectivity so far, the bulk synaptic, we haven't yet analyzed to a degree, but what I've seen, the synapse, for example, between rods and photoreceptor, between photoreceptors and bipolar cells is identical. You cannot tell apart from, if I show you an electron microscopy picture from an organoid, a human retina, whether all the connection, the specificities are the same in the human retina, unlikely, not at the detail, but we are still, this is early times. We just published this method and we are still looking into this.
Great. My next question is, it could be just a technical one. It is very interesting in these patient explorations. They were scanning the field of view. Now, this could be a simply addressable optical problem, wouldn't it, to increase the field of view so that they need to scan much less? Is it a simple, or do I just think it's simple and it's more complex than that?
Yes, of course, that's one way to go. The problem has to do with the amount of light we can project to the retina due to safety concerns. So what we are, there is a certain milliwatt per centimeter square we can project, and that is regulated by European and American agencies. It's still based on normal people. So unfortunately, we are the first to provide light to a blind person, but the regulation not yet changed. So we are sort of stuck with those regulations. So the reason we don't increase it large is that to use efficiently our photons, and that's why the patient is scanning. But now with gene therapy, we can only target a certain area in the retina. We have a certain budget of AAVs. But as you said, the best would be basically to scan it within the eye, but we don't do that because of the photon numbers.
Mm-hmm. I have a last question, which might be just a naive question. Many of the, at least two of both patients that you showed accounted for this vibrational experience.
Yeah.
What is that?
It was only one patient I showed. It was just different video, but actually all patients have it. So all patients have the vibration. So right now, my guess is the following. What is the camera doing is that when there is a change of contrast, then it shows a flash. And so if you scan, you get a lot of flashes. And maybe that is causing the vibration. So the ganglions as synchronized pulses are sent along the edges. And that's what our guess, the vibration is reporting. And the reason they record the vibration, I like to, because in normal vision, you don't, so if I take the same camera, the same goggle, I don't see vibration. But the difference here that we are stimulating ganglion cells, and then these sensors are much faster than the photoreceptors. So you have a much faster stimulation than you normally would have, and maybe that is leading to vibration.
Oh, I see. Very interesting.
You're missing a low pass filter from the front.
Okay, and some feedback issue. Okay, I see. Thank you so much. And Lars Brandin has another question in the meantime. The birds of prey have a similar high resolution area on their retina. Would this be a possible model system for use for development?
Excellent question. And we started to do that. Turns out to be not just the birds of prey. We started to do the experiments of birds of prey. We also, there are some other non-vertebrate species, so there are some foveated species. The problem is that we actually studied some of these, and birds and these other species have a completely different connectivity in the brain. So there's no cortex, but there are all sorts of nuclei, and the AVs behave very differently. So the problem is that although that the retina fovea comes up several times in evolution, it's only in mammals when the projection is very similar to humans, namely to the LGN, from the LGN to the cortex.
Thank you so much. We have just a couple more minutes left, but I asked my colleagues down at the auditorium in the hall. Actually, many people are watching you and I, mainly you, that is. You know, and I just want to ask my colleagues whether it is possible to have a question from the audience. Is it possible to relay to Professor Roska? I do not hear any feedback from them. So that means that I really have just a last task, just to say you thanks and express our heartfelt appreciation for this fascinating work. We wish you all the best en route to truly be able to restore vision. Good luck in your ventures, and thank you again for accepting our invitation, and thank you for this fantastic work.